Good morning, students. Today's session we are discussing about the seed, its structure and how the seeds germinate. We are going into its structure, let us see the three terms. What are those three terms? One is fruit, one is seed and the other one is grain. Okay, what is meant by fruits? Fruit is the enlarged or ripened ovary is called as fruit. Already you have studied in the last session that ripened ovary that will change into fruit. And you can see that what is the function of this fruit? To protect the seed. Now the second term is seed and it is the ripened ovules. So you can say that ripened ovule is called as the seed. And the seed pod protect the embryo. Inside the seed the embryo is present and the outer covering of the seed. Say for example this is a seed. This is a seed and you can see the outer covering of the seed which is called as seed pod. It protects the embryo what is inside the seed. And next one is called as grain, and grains are found you can see in maize, mostly monocots plant, just like maize, then jowar, wheat, rice, etc. We can see these words grains. These grains you can see here the fruit pod and ovary that are fused together, that is called a grain. And now we can, these three structure you understood what is meant by fruit, what is meant by a seed and what is meant by a grain. Now more about the seed we can see that it's a mature ovule after fertilization. So seed is the mature ovule after fertilization. The ovule will change into one seed. The mature ovule after fertilization is called as seed. It contains a tiny plant inside that one and the embryo is inside the tiny plant which is called as the embryo. The seed contains the tiny plant which is called as embryo. The embryo remains in inactive form till it gets a favorable condition. So the embryo it will be in an inactive condition till it get a favorable condition. That inactive condition otherwise called as dormancy. Then the seed also contains food material so that it can use these food materials during the time of germination. Then these seeds we can classify into two types. One is called as monocots and other one is called as dicots. The seed it is of two types based on the cotyledon. Based on the number of cotyledon, you can see it is divided into one that is monocot and the other one is called as dicot. In a seed, if only one cotyledon is present, it is called as what? Monocotyledon. And if it is two, usually you can see that maize grain, and then rice, wheat, etc. You can see here. Here is an example of maize. This contains only one cotyledon, so it's a monocot. Whereas this is a bean seed. This is a bean seed which contains two cotyledons. So if the seed contains only one cotyledon, it is called a monocotyledon, and if it contains two cotyledons, it is called as dicotyledon. And again now we can see that seeds may vary in their size, different sizes of the seeds. Some seeds are very small in size, example poppy seeds or orchid seeds, these are very small in size. Some are bigger, some other seeds are bigger in size. Say for example pumpkin, watermelon, etc. You can see some more large size seed. And still again in a mango seed you can see its size is some more bigger. And still you can see the largest seed, it is the coconut seed. It is about big in size. Then the size, shape and structure of the seeds, different plants vary considerably on the basis of the structure of the mold, but on the structure is mostly same. Now on the basis of endosperm present in a seed, it is divided into two. One is called as albuminous, means it has endosperm which is otherwise called as endospermic seed, and other one is exalbuminous. And this you can see that cotyledon store the food materials in the exalbuminous or non-endospermic seed. Now we can go to the structure of a bean seed. What are the things you can see? So there is an outer covering of the seed which is called a seed coat. And this seed coat, externally two layers are there. One is called as testa and the other one is called as tegmen. So a bean seed I am drawing here. Almost the shape of a kidney. It is the shape of a kidney, and you can see this is what is called as testa, and inside that is still again another more layer like this, and this is called as tegmen. Both this testa and tegmen, what is the function? It protects the seed. Usually this is present in the outside. So you see this bean seed has an outer convex side and inner concave side is there. So this both structure, the testa and tegmen, which are the two layers of seed coat, that will protect the seed. That is the main function of this testa and tegmen. Then next you can see, if you observe a seed, you can see a white structure. Here you see the concave side of the seed. You can see a white structure, small white structure, which is called as hilum. Here there is a white structure which is called as hilum. This is the structure called as hilum, and you can see that this is the spots where these ovules are attached to the ovarian wall, or you can see that with the help of placenta, this region, this is the hilum region, it is attached to the ovarian wall inside when it is in the inside condition. Next one, you can see that there is a small opening. Here there is a small opening. This is called as micropyle. Micropyle, a small opening below the cotyledons, you can see, which is called as micropyle. And what is its function? Let us see. One is to take water. You can see when you soak a seed in the water, you can see it will absorb water and it swells up, and through which part, mainly through the micropyle, water absorption takes place. Again you can see that it provides the diffusion of respiratory gases. Which is the respiratory gases of oxygen? Yes, you can see it provides for the diffusion of respiratory gases for the growth of the embryo. Below the seed coat there are two cotyledons here in the bean seed. So these are the two cotyledons. Okay, one if you are showing. This is another cotyledon. So here you can see another cotyledon. So dicotyledon, two here one cotyledon, this is another cotyledon. So below the micropyle you can see two cotyledons are there, and inside the cotyledon, careful observation, you can see inside that, if you are opening that one, you can see a small embryo, a tiny embryo that can be seen easily attached to this one. See. Here, if you cut open this one, inside this here, you can see a small embryo is present attached to the cotyledons. The embryo consists of two parts. One is called as the radicle and the other one is called as plumule. So, two parts, the embryo consists of two parts. One is called as radicle and the other one is called as plumule. that it grows and change into roots. And whereas plumule, it becomes the shoot system and the leaves will be forming from that one. Here you can say, here it will be attached and this will be the radicle, and here small leaf-like structure will be forming. This is the plumule. This is the radicle and this is the plumule. So the embryo inside the cotyledon, it consists of plumule and the radicle. Plumule will change in the shoot system. Radicle will change into the root system. So again we can go to another monopod seed, that is maize. Maize, you are familiar. These are some of the maize seeds I am showing you. These are the maize seeds. And maize is actually a one-seeded fruit in which fruit ball and seed coat are fused together. So we are not calling it as a seed or a fruit, we are calling it as a grain. Maize, why we are calling it as a grain? Because here the fruit ball and the seed coat are fused together, and so we are calling it as a grain. And you can see that on one side of the grain occurs a small light covered oval area. See, this is the structure of the grain. See, maize grain, and you can see an oval here. One area is like this. Here, this is the embryo. You can see that embryo is downward, and here this is the large endosperm. The yellow color, which is the large endosperm, and the white color you can see here, that is the embryo. And the embryonic parts and this, you can say, endosperm, both are separated by a layer. See, if you are drawing, it will be like this, and this region, this is a large endosperm present. And this is the endosperm. And this region together we can say that this is the embryonic part. And here you can see that this is separated, this endosperm and as well as this embryo part is separated by a layer, and it is called as epithelial layer is there, and on the top of that this is rich in protein, and this side it is called as a neuron. A neuron layer. So here we can see that clearly in this seed. You can see close observation. If you are taking a maize seed and looking, you can clearly notice all these things. And the embryo consists of a single cotyledon. As the maize grain, it is having only one cotyledon. It is a monocotyledon, and here it is called as scutellum. So this region it is called as scutellum. It consists of only one cotyledon, and so it is called as scutellum. And it contains both the radical region as well as the plumule region. Here downward you can see this radical region, and here you can see the plumule region. And the radicle, it has a protective covering called as polyorhiza, and so that is here the polyorhiza and here the protective region of the plumule is called as polyoctile. So polyorhiza, polyoctile, these are the two protective regions of the radicle as well as plumule. So that's all about the structure of the maize grain. I think you got it, the differences between a diploid seed as well as a monopod seed. Once we can summarize, that is, here we have taken the example bean seed as well as maize grain, and in the case of beans, that is, two cotyledon is present, whereas in the case of a maize grain only one cotyledon is present. And here no endosperm in the first I am telling about bean, next about maize grain. Here no endosperm, whereas maize grain large endosperm is present. Then here large embryo is present, and whereas in the maize you can see small embryo is present. And in the case of bean seeds, plumule leaves are folded, whereas here plumule leaves are folded in condition. Next one, hilum and micropyle are visible in the case of bean seed, whereas hilum and micropyle are not visible in the case of a maize grain. Then seed separately contained in the fruit called pod. So this is with these bean seeds and all. Outer one green covering you can say that is what is called as pod. Whereas here there is a single grain and no pod is present. That's all about bean seed as well as a maize grain, which is one is the dicot one and the other one is a monocot one. Now we can see the germination, how germination takes place in a plant. The seed contains a dormant embryo. I already told you what is meant by dormant. You can see when we are taking a dry seed and looking, you will notice that as if it is dead. Okay, because it is dried one, but still inside the seed certain process is going on. This is of the food like that, certain process is going on, but externally we cannot say anything. So this is called as dormant embryo. When this dormant embryo get a favorable condition, you can say it starts to grow. The small seedling start to grow from that seed, and that process is called as what? Germination. So germination is the process of formation of a seedling from the embryo. So germination is the process of formation of a small seedling from the embryo. Now let us see what are the conditions necessary for this germination. In order to germinate a seed, what are the three conditions necessary? First one is water. Let us see how water helps in germination. So, the seed obtains water from the environment. That is usually from the, so when we sow the seed in the soil, which is the environment in which it gets water. Yes, it gets water from the soil. And you can see that it imbibes water and it swells. Through which part of the seed it will absorb water? Yes, through the micropyle, the water will be absorbed by the seed and it swells up. And what is necessary for the chemical reaction and for the enzymes to that act upon the stored in the cotyledons for the endosperm. So, water is very necessary for germination. In the absence of the water, seeds may not germinate. Let us see the next condition is suitable temperature. Second one is suitable suitable temperature is needed for the growth of this seed. If it is too cold, you can see that what will happen, the growth will be inhibited. No growth will take place, and if the more temperature, okay, decay of the cell also or spoilage of the cell will be taking place. It destroys the growth of the cell. So there is a temperature, a minimum or a maximum temperature is needed. That is usually 25 degrees Celsius to 35 degrees Celsius. So that the suitable temperature is between 25 degree to 35 degrees Celsius is needed for the proper germination of the seed, and this temperature is called as optimum temperature. So the optimum temperature for the germination of the seed is 25 to 35 degrees Celsius. Next favorable condition needed for the germination of the seed is oxygen. So here you can see that the respiratory gas is oxygen. The seed want to respire, and for the rapid cell division also, and for the growth also it need what energy it is needed. From where this energy is getting? Yes, by the respiration. So oxygen is also needed for respiration. So these are the three main conditions necessary for germination of a seed. The first one is water, second one is suitable temperature, that is between 25 degree to 35 degrees Celsius, and the last one is oxygen. Now you may get a wonderful question that the seeds grown deep in the soil. You can see that we are sowing the seed in the soil. If it is in the correct level only, that correctly germination takes place. If it goes deep down, the seeds may not germinate. What's the reason for this thing? Yes, proper supply of oxygen will not get, the seed will not get proper oxygen for respiration, so that cell division will not take place. And again, insufficient pushing force. The seed want to germinate, you can see that it's a epicotyl, hypocotyl, that it has to come out, isn't it? So the insufficient pushing force will not be available for the embryo. So because of these two reasons, you can see that seeds sown in deep soil fail to germinate. Now we can see some experiments to prove that these three conditions, that is water, suitable temperature and oxygen, these three conditions are necessary for the proper germination of a seed. Now first let us see that an experiment to prove that water is necessary for germination. So you have to take some seeds, say for example any green gram or bean seeds, any type of seeds you are taking, and 5 seeds you are keeping in a cotton wool, kept it like that, dry cotton wool. There are another 5 seeds in a wet cotton wool, that is you sprinkle water on that cotton and then you are keeping in that one. Keep it for 2 or 3 days like that. One change you can see, which one will germinate and which one will not germinate. Yes, the first one, of course, there is no availability of water. So will it germinate? No, it won't germinate. Whereas second one, you have sprinkled water on the cotton wool so that you can clearly see that it will be germinating. This simple experiment shows that what is necessary for germination. Now the second experiment to prove that suitable temperature, that is what is the optimum temperature is needed? Yes, from 25 degree to 35 degree. This optimum temperature is needed for the growth of, for germinating the seed. As in the same case, you have to take 2 beakers. In that one you keep like that cotton wool you have to keep, and few seeds you have to keep in that one. Second one also the cotton wool you have to take and sprinkle it with water, and then one keep outside in the normal room temperature and the other one you keep it in a refrigerator. You can see after 2-3 days what you kept in the normal temperature, that is in the room temperature, it starts to germinate, and the other one which you kept inside the refrigerator, it has not started its germination. Why it happens? Yes, inside this refrigerator, why germination does not take place? Because yes, the temperature is too low in the inside the refrigerator. Whereas in the room, you can see normal temperature the seed is getting. So seeds started to germinate. So this experiment, this is also another simple experiment which shows that a suitable temperature is needed for the seeds to germinate. Third one is experiment to prove that oxygen is necessary for germination. How you can do this is also another simple experiment. You can see that you take in a 2 conical flask. You have to take the same seeds in one cotton wool. You both it should be wet, and you can see that in one of the conical flask you keep the seed and you sprinkle the water on that one. That is cotton wool should be get wet. And you have to keep a test tube inside that one. This is a conical flask. Here, inside the cotton wool, you have to take the seeds like this, and here, this is the here using a thread. You have to insert a test tube here, and this test tube contains normal water. Okay. In the same way, in the second case, in the here you are taking the seed, and in the test tube here using a thread you can tie it. A test tube you are taking here, and in that you have to take alkaline pyrogallic acid. What is the speciality of this pyrogallic acid? Yes, it will absorb what any oxygen is present. It will absorb that pyrogallic acid. Oxygen it will absorb, and keep the two apparatus here. You can see that two apparatus also you have to keep like this, and meanwhile after 2-3 days you can see that the first conical flask which containing the seed start to germinate, and whereas in the second one that seeds will not germinate. Why seed will not germinate? Yes, because it is not getting oxygen. Why it is not getting oxygen? Because in a test tube we have kept what as alkaline pyrogallic acid, and this will absorb, this chemical will absorb what? If any oxygen is present, it will absorb that oxygen, so the seeds will not get oxygen. So that seed will not germinate. So with these three simple experiments, first case we have proved that what is necessary for germination. Second case we have proved that what optimum temperature, that is 25 to 35 degrees Celsius is needed for germination, and in the third case we have proved that oxygen is needed for germination. Now with in a single experiment the three conditions also we can prove. Let us go to that experiment. You have to take a beaker like this, half of it should be filled with water, and then you have to take a glass slide like this. This is a glass slide you have seen in biology lab. Okay, three seeds you have to tie here. First seed here. Okay, second seed in the middle, and the third seed here. Okay, tie this using a thread. You can tie this one. Okay, each one you can tie. Now, what you have to do is this glass slab. After tying this one, you have to immerse it in this water. Okay, so the first seed will be here. Second seed will be here, and the third seed will be here. See the position of the seed. First seed it is completely immersed in water. Second seed you can see that it is just above just below the water level, and a little part you can see upside. Whereas the third seed you can see it is completely above the water, no contact with the water. Keep it aside like this for 2 or 3 days. After 3 days, when you are observing, you can see which seed will germinate. Can you say? Whether it is the first one, or the second one, or the third one. Yes, the second seed will be germinating. So here you can see that it slowly starts to germinate. More leaves are coming in the second seed. Why it happens so? Why germination does not take place in these both seeds? Yes, we can see that the first seed it is completely under the water. It is getting the condition water it is getting, whereas it is not getting oxygen. So this seed will not germinate. Whereas the third seed you can see that it is getting oxygen, whereas it is not getting water. So there is no probability. Whereas here the middle seed or the second seed it starts to germinate, it gets water as well as oxygen. So this proves that oxygen, this water as well as oxygen is very much important in the case of germination. Now let us see what are the types of germination. These two terms, epi and hypo, the two types of germination. One is called as hypogeal germination and the other one is called as epigeal germination. So with the examples we are studying about epigeal germination as well as the hypogeal germination. The region of the axis between the point of attachment of the cotyledon and the plumule is called as epicotyl. And what is meant by hypocotyl? The region of the axis below the cotyledon is called as hypocotyl. So, below the cotyledon, what region is called as hypocotyl, and above that region is called as what? Epicotyl. Based on this, you can see that two types of germination. One is called as epigeal germination and the other one is hypogeal germination. You can see that if the epicotyl elongates, the cotyledon remain underground, and you can see that is called as hypogeal germination. And in the case of pea plant and all, that is below the cotyledon, during the time of germination, cotyledon will be below the soil, and again you can see that hypogeal germination, epigeal germination, you can see that cotyledons come above the soil. That is the two type of germination. Now let us see that two differences between hypogeal germination and epigeal germination. Cotyledon remains in the underground. So you can see that in the case of hypogeal germination, at the time of germination, cotyledons will be below the soil. Whereas in epigeal germination, cotyledons will come above the soil. Next, epicotyl elongates faster in the case of hypogeal germination, whereas in the case of epigeal germination, hypocotyl elongates faster. And third one, usually in hypogeal germination, you can see that usually occurs in monocots plant, whereas this epigeal germination that occurs in dicots plant. So these are the main differences between the epigeal germination and the hypogeal germination. And now let us see some germination taking how in which plant this hypogeal germination takes place and in which plant epigeal germination takes place. So first we can see that we are taking the peas, green pea seed we are taking, and how this germination takes place. And pea seeds, the seed absorb water and cells up. You can see if we put inside the soil from which is in its environment, that is from the soil. First thing is that it absorbs water and then it swells up. Next, what will happen? The seed coat will burst. The seed coat will be break open and the radicle emerges out. Radicle means root. That will be changing in the root. The radicle emerges out downward and form the root system, and the plumule grows up and form the shoot system. So let us see, this is the soil level. The seed we are sowing in the pea seed we are sowing in the soil, and you can see that it will absorb the water, and you can see little bit size will be increasing, that is swelling of the seed takes place. And next step you can see that as the swelling takes place, seed pod will rupture, it will break open, and the radicle slowly you can see that root, that is radicle will emerge out, and next plumule. Here with the small leaf it will be coming out, and in the next stage you can see that this will be remaining like this only in the soil only, and slowly only the plumule which grows from here, it grows, this is the soil level, it grows above the soil, okay. And so where will be the cotyledon at the time of germination? Yes, it is below the soil. So in the case of pea and most of the monocots plant also you can see that the seed will be remaining inside the soil or below the soil at the time of germination, and such type of germination we are calling as hypogeal germination. Now going to the bean seed, now let us see how it takes place. Yes, here also the water it will absorb bean seed. The same way you can see this is the soil level, and bean seeds we are putting in the soil, and it again you can see absorb water from the environment and it swells up, and here also you can see that seed pod breaks open. What are the two seed pod, that is testa and tegmen, it breaks open, and next you can see that like this it will be coming up in the cotyledon will come. Yes, it is epithelial germination takes place and the cotyledon comes above the soil. So first step, you can see seed will be sown in the soil and it imbibe water and it swells up. Seed pod ruptures and the root will be small radicle will be coming up. At the same time, plumule also which pushes this cotyledons up in the soil. So again you can see in the third stage, totally radicle will go down and this cotyledon will come above the soil. So this step is called as epithelial germination. And in the case of maize grain, say again hypogeal germination only because it is a monocotyledon seed. There also you can see that seed will be remaining inside the soil and above the soil. During the time of germination, the cotyledon will not come because since it is a monocotyledon plant. Usually in almost all monocotyledon plant we can see hypogeal germination only, that is cotyledon will not appear above the soil. Now another type of germination is the which is called as viviparous germination, that is usually in rhizopora, a plant. When the seed is attached to the plant itself, it will start to germinate, and then you can see after this root system and all come, you can see that seed will be germinating, radicle will come, and slowly small plumule also will start to come, and then you can see that this germination takes place. So that is called as viviparous germination. And small seedling, germination of the after seedling, you can see with the help of root, it will attaches or fixes to the soil, and the shoot system grow above the ground and small leaves will be coming. Then it starts to synthesize its own food by the process called as photosynthesis. Then each case you can see cell will divide and redivide, and the growth will take place. Branches also will be forming in this plant, and thus these plants will become big. Later flowers will be formed. Again from the flower seed will be formed, and from these seeds a new plant will be coming out. So that's all about this seeds and its germination. You have seen the structure of the seed as well as the germination. Two types of germination, that is hypogeal germination, which is below the soil, cotyledon remains below the soil, and epithelial germination, cotyledon appears above the soil. That's all about this lesson.